Brought to you by WikiVD Documentaries. Fungus. A fungus is any member of the group of eukaryotic organisms that includes microorganisms such as yeasts and molds, as well as the more familiar mushrooms. These organisms are classified as a kingdom, fungi, which is separate from the other eukaryotic life kingdoms of plants and animals. A characteristic that places fungi in a different kingdom from plants, bacteria, and some protists is chitin in their cell walls. Similar to animals, fungi are heterotrophs. They acquire their food by absorbing dissolved molecules, typically by secreting digestive enzymes into their environment. Fungi do not photosynthesize. Growth is their means of mobility, except for spores, which may travel through the air or water. Fungi are the principal decomposers in ecological systems. These and other differences place fungi in a single group of related organisms, named the Eumycota, which share a common ancestor, an interpretation that is also strongly supported by molecular phylogenetics. This fungal group is distinct from the structurally similar myxomycetes and omycetes. The discipline of biology devoted to the study of fungi is known as mycology. In the past, mycology was regarded as a branch of botany. Although it is now known fungi are genetically more closely related to animals than to plants. Abundant worldwide, most fungi are inconspicuous because of the small size of their structures and their cryptic lifestyles in soil or on dead matter. Fungi include symbionts of plants, animals, or other fungi and also parasites. They may become noticeable when fruiting, either as mushrooms or as molds. Fungi perform an essential role in the decomposition of organic matter and have fundamental roles in nutrient cycling and exchange in the environment. They have long been used as a direct source of human food, in the form of mushrooms and truffles, as a leavening agent for bread, and in the fermentation of various food products, such as wine, beer, and soy sauce. Since the 1940s, fungi have been used for the production of antibiotics, and more recently, various enzymes produced by fungi are used industrially and in detergents. Fungi are also used as biological pesticides to control weeds, plant diseases, and insect pests. Many species produce bioactive compounds called mycotoxins, such as alkaloids and polyketides, that are toxic to animals, including humans. The fruiting structures of a few species contain psychotropic compounds and are consumed recreationally or in traditional spiritual ceremonies. Fungi can break down manufactured materials and buildings, and become significant pathogens of humans and other animals. Losses of crops due to fungal diseases or food spoilage can have a large impact on human food supplies and local economies. The fungus kingdom encompasses an enormous diversity of taxa, with varied ecologies, life cycle strategies, and morphologies ranging from unicellular aquatic chytrids to large mushrooms. However, little is known of the true biodiversity of kingdom fungi, which has been estimated at 1.5 million to 5 million species, with about 5% of these having been formally classified ever since the pioneering 18th and 19th century taxonomical works of Carl Linnaeus, Christian Hendrik Persoon, and Elias Magnus Fries. Fungi have been classified according to their morphology of physiology. Advances in molecular genetics have opened the way for DNA analysis to be incorporated into taxonomy, which has sometimes challenged the historical groupings based on morphology and other traits. Phylogenetic studies published in the last decade have helped reshape the classification within kingdom fungi, which is divided into 1 subkingdom, 7 phyla, and 10 subphyla. Etymology. The English word fungus is directly adopted from the Latin fungus, used in the writings of Horace and Pliny. This in turn is derived from the Greek words fungos, which refers to the macroscopic structures and morphology of mushrooms and molds. The root is also used in other languages, such as the German Schwamm and Schimmel. The use of the word mycology, which is derived from the Greek mykes and logos, to denote the scientific study of fungi, is thought to have originated in 1836, with English naturalist Miles Joseph Berkeley's publication The English Flora of Sir James Edward Smith, Volume 5, a group of all the fungi present in a particular area or geographic region is known as Mycobiota, and more, the Mycobiota of Ireland. Characteristics. Before the introduction of molecular methods for phylogenetic analysis, taxonomists considered fungi to be members of the plant kingdom. Because of similarities in lifestyle, both fungi and plants are mainly immobile, and have similarities in general morphology and growth habitat. Like plants, fungi often grow in soil and, in the case of mushrooms, form conspicuous fruit bodies, which sometimes resemble plants such as mosses. The fungi are now considered a separate kingdom, distinct from both plants and animals, from which they appear to have diverged around 1 billion years ago. Some morphological, biochemical, and genetic features are shared with other organisms, while others are unique to the fungi, clearly separating them from the other kingdoms. Shared features, unique features. Most fungi lack an efficient system for the long-distance transport of water and nutrients, such as the xylem and phloem in many plants. To overcome this limitation, some fungi, such as Armillaria, form rhizomorphs, which resemble and perform functions similar to the roots of plants. As eukaryotes, fungi possess a biosynthetic pathway for producing terpenes that uses mevalonic acid and pyrophosphate as chemical building blocks. Plants and some other organisms have an additional terpene biosynthesis pathway in their chloroplasts, a structure fungi and animals do not have. Fungi produce several secondary metabolites that are similar or identical in structure to those made by plants. Many of the plant and fungal enzymes that make these compounds differ from each other in sequence and other characteristics, which indicate separate origins and convergent evolution of these enzymes in the fungi and plants. Diversity. Fungi have a worldwide distribution, and grow in a wide range of habitats, including extreme environments such as deserts or areas with high salt concentrations, or ionizing radiation, as well as in deep-sea sediments. Some can survive the intense UV and cosmic radiation encountered during space travel. Most grow in terrestrial environments, though several species live partly or solely in aquatic habitats, such as the chytrid fungus Batrachochytrium dendrobatidis, a parasite that has been responsible for a worldwide decline in amphibian populations. This organism spends a part of its life cycle as a motile zoospore, enabling it to propel itself through water and then to its amphibian host. Other examples of aquatic fungi include those living in hydrothermal areas of the ocean. Around 100,000 species of fungi have been formally described by taxonomists, but the global biodiversity of the fungus kingdom is not fully understood. On the basis of observations of the ratio of the number of fungal species to the number of plant species in selected environments, the fungal kingdom has been estimated to contain about 1.5 million species. A recent estimate suggests there may be over 5 million species. In mycology, species have historically been distinguished by a variety of methods and concepts. Classification based on morphological characteristics, such as the size and shape of spores or fruiting structures, has traditionally dominated fungal taxonomy. Species may also be distinguished by their biochemical and physiological characteristics, such as their ability to metabolize certain biochemicals, or their reaction to chemical tests. The biological species concept discriminates species based on their ability to mate. The application of molecular tools, such as DNA sequencing and phylogenetic analysis, to study diversity has greatly enhanced the resolution and added robustness to estimates of genetic diversity within various taxonomic groups. Fungi has 36,000 or more sexes. Most species have only 2 sexes. Mycology. Mycology is the branch of biology concerned with the systematic study of fungi, including their genetic and biochemical properties, their taxonomy, and their use to humans as a source of medicine, food, and psychotropic substances consumed for religious purposes, as well as their dangers, such as poisoning or infection. The field of phytopathology, the study of plant diseases, is closely related, because many plant pathogens are fungi. The use of fungi by humans dates back to prehistory. See the Iceman. A well-preserved mummy of a 5,300-year-old Neolithic man found frozen in the Austrian Alps carried 2 species of polypore mushrooms that may have been used as tinder, or for medicinal purposes. Ancient peoples have used fungi as food sources, often unknowingly, for millennia, in the preparation of leavened bread and fermented juices. Some of the oldest written records contain references to the destruction of crops that were probably caused by pathogenic fungi. History. Mycology is a relatively new science that became systematic after the development of the microscope in the 17th century. Although fungal spores were first observed by Giambattista della Porta in 1588, the seminal work in the development of mycology is considered to be the publication of Pier Antonio Michelis' 1729 work Nova Plantarum Genera. Micheli not only observed spores, but also showed that, under the proper conditions, they could be induced into growing into the same species of fungi from which they originated, extending the use of the binomial system of nomenclature introduced by Carl Linnaeus in his Species Plantarum. The Dutch Christian Hendrik Persoon established the first classification of mushrooms with such skill so as to be considered a founder of modern mycology. Later, Elias Magnus Fries further elaborated the classification of fungi, using spore color and various microscopic characteristics, methods still used by taxonomists today. Other notable early contributors to mycology in the 17th, 19th, and early 20th centuries include Miles Joseph Barclay, August Carl Joseph Corder, Anton de Barry, the brothers Louis René and Charles Toulon, Arthur H. R. Buller, Curtis G. Lloyd, and Pier Andrea Saccardo. The 20th century has seen a modernization of mycology that has come from advances in biochemistry, genetics, molecular biology, and biotechnology. The use of DNA sequencing technologies and phylogenetic analysis has provided new insights into fungal relationships and biodiversity, and has challenged traditional morphology-based groupings in fungal taxonomy. Microscopic structures. Most fungi grow as hyphae, which are cylindrical, thread-like structures 2 to 10 micrometers in diameter, and up to several centimeters in length. Hyphae grow at their tips. New hyphae are typically formed by emergence of new tips along existing hyphae. by a process called branching, or occasionally growing hyphal tips fork, giving rise to two parallel growing hyphae. Hyphae also sometimes fuse when they come into contact, a process called hyphal fusion. These growth processes lead to the development of a mycelium, an interconnected network of hyphae. Hyphae can be either septate or coenocytic. Septate hyphae are divided into compartments separated by cross walls, with each compartment containing one or more nuclei. Coenocytic hyphae are not compartmentalized. Septa have pores that allow cytoplasm, organelles, and sometimes nuclei to pass through. An example is the dolopore septum in fungi of the phylum Basidiomycota. Coenocytic hyphae are in essence multinucleate supercells. Many species have developed specialized hyphal structures for nutrient uptake from living hosts. Examples include Haustoria in plant parasitic species of most fungal phyla, and arbuscules of several mycorrhizal fungi, which penetrate into the host cells to consume nutrients. Although fungi are opisthokonts, a grouping of evolutionarily related organisms broadly characterized by a single posterior flagellum, all phyla except for the chytrids have lost their posterior flagella. Fungi are unusual among the eukaryotes in having a cell wall that, in addition to glucans and other typical components, also contains the biopolymer chitin. Macroscopic structures. Fungal mycelia can become visible to the naked eye, for example, on various surfaces and substrates, such as damp walls and spoiled food, where they are commonly called molds. Mycelia grown on solid agar media in laboratory Petri dishes are usually referred to as colonies. These colonies can exhibit growth shapes and colors that can be used as diagnostic features in the identification of species or groups. Some individual fungal colonies can reach extraordinary dimensions and ages, as in the case of a clonal colony of Ulmalaria solidipes, which extends over an area of more than 900 hectares, with an estimated age of nearly 9,000 years. The apothecium, a specialized structure important in sexual reproduction in the ascomycetes, is a cup-shaped fruit body that is often macroscopic and holds the hymenium, a layer of tissue containing the spore-bearing cells. The fruit bodies of the basidiomycetes and some ascomycetes can sometimes grow very large, and many are well known as mushrooms. Growth and physiology. The growth of fungi as hyphae on or in solid substrates, or as single cells in aquatic environments, is adapted for the efficient extraction of nutrients. Because these growth forms have high surface area to volume ratios, hyphae are specifically adapted for growth on solid surfaces, and to invade substrates and tissues. They can exert large penetrative mechanical forces. For example, many plant pathogens, including magnopod grisi, form a structure called an oppressorium, that evolved to puncture plant tissues. The pressure generated by the oppressorium, directed against the plant epidermis, can exceed the filamentous fungus Psilomyces lilacinus uses a similar structure to penetrate the eggs of nematodes. The mechanical pressure exerted by the oppressorium is generated from physiological processes that increase intracellular turgor by producing osmolytes such as glycerol. Adaptations such as these are complemented by hydrolytic enzymes secreted into the environment to digest large organic molecules, such as polysaccharides, proteins, and lipids, into smaller molecules that may then be absorbed as nutrients. The vast majority of filamentous fungi grow in a polar fashion by elongation at the tip of the hypha. Other forms of fungal growth include intercalary extension, as in the case of some endophytic fungi, or growth by volume expansion during the development of mushroom stipes and other large organs. Growth of fungi as multicellular structures consisting of somatic and reproductive cells, a feature independently evolved in animals and plants, has several functions, including the development of fruit bodies for dissemination of sexual spores and biofilms for substrate colonization and intercellular communication. The fungi are traditionally considered heterotrophs, organisms that rely solely on carbon fixed by other organisms for metabolism. Fungi have evolved a high degree of metabolic versatility that allows them to use a diverse range of organic substrates for growth, including simple compounds such as nitrate, ammonia, acetate, or ethanol. In some species, the pigment melanin may play a role in extracting energy from ionizing radiation, such as gamma radiation. This form of radiotrophic growth has been described for only a few species. The effects on growth rates are small, and the underlying biophysical and biochemical processes are not well known. This process might bear similarity to CO2 fixation via visible light, but instead uses ionizing radiation as a source of energy. Reproduction. Fungal reproduction is complex, reflecting the differences in lifestyles and genetic makeup within this diverse kingdom of organisms. It is estimated that a third of all fungi reproduce using more than one method of propagation. For example, reproduction may occur in two well-differentiated stages within the life cycle of a species: the teleomorph and the anamorph. Environmental conditions trigger genetically determined developmental states that lead to the creation of specialized structures for sexual or asexual reproduction. These structures aid reproduction by efficiently dispersing spores or spore-containing propagules. Spore dispersal. Both asexual and sexual spores or sporangiospores are often actively dispersed by forcible ejection from their reproductive structures. This ejection ensures exit of the spores from the reproductive structures as well as traveling through the air over long distances. Specialized mechanical and physiological mechanisms, as well as spore surface structures, enable efficient spore ejection. For example, the structure of the spore-bearing cells in some ascomycete species is such that the buildup of substances affecting cell volume and fluid balance enables the explosive discharge of spores into the air. The forcible discharge of single spores, termed ballistospores, involves formation of a small drop of water, which upon contact with the spore leads to its projectile release, with an initial acceleration of more than 10,000 grams. The net result is that the spore is ejected 0.01–0.02 cm, sufficient distance for it to fall through the gills or pores into the air below. Other fungi, like the puffballs, rely on alternative mechanisms for spore release, such as external mechanical forces. The bird's nest fungi use the force of falling water drops to liberate the spores from cup-shaped fruiting bodies. Another strategy is seen in the stinkhorns, a group of fungi with lively colors and putrid odor that attract insects to disperse their spores. Evolution. In contrast to plants and animals, the early fossil record of the fungi is meager. Factors that likely contribute to the underrepresentation of fungal species among fossils include the nature of fungal fruiting bodies, which are soft, fleshy, and easily degradable tissues, and the microscopic dimensions of most fungal structures, which therefore are not readily evident. Fungal fossils are difficult to distinguish from those of other microbes, and are most easily identified when they resemble extant fungi. Often recovered from a permineralized plant or animal host, these samples are typically studied by making thin section preparations that can be examined with light microscopy or transmission electron microscopy. Researchers study compression fossils by dissolving the surrounding matrix with acid and then using light or scanning electron microscopy to examine surface details. The earliest fossils possessing features typical of fungi date to the Paleoproterozoic era. These multicellular benthic organisms had filamentous structures capable of anastomosis. Other studies estimate the arrival of fungal organisms at about 760–1060 Ma on the basis of comparisons of the rate of evolution in closely related groups. For much of the Paleozoic era, the fungi appear to have been aquatic and consisted of organisms similar to the extant chytrids in having flagellum-bearing spores. The evolutionary adaptation from an aquatic to a terrestrial lifestyle necessitated a diversification of ecological strategies for obtaining nutrients, including parasitism, saprobism, and the development of mutualistic relationships such as mycorrhiza and lichenization. Recent studies suggest that the ancestral ecological state of the Ascomycota was saprobism, and that independent lichenization events have occurred multiple times. It is presumed that the fungi colonized the land during the Cambrian, long before land plants. Fossilized hyphae and spores recovered from the Ordovician of Wisconsin resemble modern-day glomerales, and existed at a time when the land flora likely consisted of only non-vascular bryophyte-like plants. Prototaxotes, which was probably a fungus or lichen, would have been the tallest organism of the late Silurian. Fungal fossils do not become common and uncontroversial until the early Devonian, when they occur abundantly in the Rhynie chert, mostly as Zygomycota and Chytridiomycota. At about this same time, approximately 400 Ma, the Ascomycota and Basidiomycota diverged, and all modern classes of fungi were present by the late Carboniferous. Lichen-like fossils have been found in the Dushantuo Formation in southern China dating back to 635,551 Ma. Lichens formed a component of the early terrestrial ecosystems, and the estimated age of the oldest terrestrial lichen fossil is 400 Ma. This date corresponds to the age of the oldest known sporocarp fossil, a Paleoperennomycete species found in the Rhynie chert. The oldest fossil with microscopic features resembling modern-day Basidiomycetes is Paleoancestris, found permineralized with a fern from the Pennsylvanian. Rare in the fossil record are the Homo Basidiomycetes. Two amber-preserved specimens provide evidence that the earliest known mushroom-forming fungi appeared during the late Cretaceous, 90 Ma. Some time after the Permian Triassic extinction event, a fungal spike formed, suggesting that fungi were the dominant life form at this time, representing nearly 100% of the available fossil record for this period. However, the relative proportion of fungal spores relative to spores formed by algal species is difficult to assess. The spike did not appear worldwide, and in many places it did not fall on the Permian-Triassic boundary. Taxonomy. Although commonly included in botany curricula and textbooks, fungi are more closely related to animals than to plants and are placed with the animals in the monophyletic group of Opisthokonts. Analyses using molecular phylogenetics support a monophyletic origin of the fungi. The taxonomy of the fungi is in a state of constant flux, especially due to recent research based on DNA comparisons. These current phylogenetic analyses often overturn classifications based on older, and sometimes less discriminative methods based on morphological features and biological species concepts obtained from experimental matings. There is no unique, generally accepted system at the higher taxonomic levels, and there are frequent name changes at every level from species upwards. Efforts among researchers are now underway to establish and encourage usage of a unified and more consistent nomenclature. Fungal species can also have multiple scientific names depending on their life cycle and mode of reproduction. Websites such as Index Fungorum and ITIS list current names of fungal species. The 2007 classification of kingdom fungi is the result of a large-scale collaborative research effort involving dozens of mycologists and other scientists working on fungal taxonomy. It recognizes 7 phyla, 2 of which, the Ascomycota and the Basidiomycota, are contained within a branch representing subkingdom Dicaria. The most species-rich and familiar group, including all the mushrooms, most food spoilage molds, most plant pathogenic fungi, and the beer, wine, and bread yeasts. The accompanying cladogram depicts the major fungal taxa and the relationship to Opisthoco and uniconed organisms, based on the work of Philippe Seiler and the Mycota, a comprehensive treatise on fungi as experimental systems for basic and applied research. The lengths of the branches are not proportional to evolutionary distances. Taxonomic groups. The major phyla of fungi have been classified mainly on the basis of characteristics of their sexual reproductive structures. Currently, 7 phyla are proposed: Microsporidia, Chytridiomycota, Blasticladiomycota, Neocorlomystigomycota, Glomeromycota, Ascomycota, and Basidiomycota. Phylogenetic analysis has demonstrated that the Microsporidia, unicellular parasites of animals and protists, are fairly recent and highly derived endobiotic fungi. One 2006 study concludes that the Microsporidia are a sister group to the true fungi, that is, they are each other's closest evolutionary relative. Hibbett and colleagues suggest that this analysis does not clash with their classification of the fungi. And although the Microsporidia are elevated to phylum status, it is acknowledged that further analysis is required to clarify evolutionary relationships within this group. The Chytridiomycota are commonly known as chytrids. These fungi are distributed worldwide. Chytrids and their close relatives Neocorlomystigomycota and Blasticladiomycota are the only fungi with active motility, producing zoospores that are capable of active movement through aqueous phases with a single flagellum, leading early taxonomists to classify them as protists. Molecular phylogenies, inferred from rRNA sequences in ribosomes, suggest that the chytrids are a basal group divergent from the other fungal phyla, consisting of 4 major clades with suggestive evidence for paraphyly, possibly polyphyly. The Blasticladiomycota were previously considered a taxonomic clade within the Chytridiomycota. Recent molecular data and ultrastructural characteristics, however, place the Blasticladiomycota as a sister clade to the Zygomycota, Glomeromycota, and Dicaria. The Blasticladiomycetes are saprotrophs, feeding on decomposing organic matter, and they are parasites of all eukaryotic groups. Unlike their close relatives, the chytrids, most of which exhibit zygotic meiosis, the Blasticladiomycetes undergo sporic meiosis. The Neocorlomystigomycota were earlier placed in the phylum Chytridomycota. Members of this small phylum are anaerobic organisms, living in the digestive system of larger herbivorous mammals and in other terrestrial and aquatic environments enriched in cellulose. They lack mitochondria, but contain hydrogenosomes of mitochondrial origin, as in the related chytrids. Neocorla mastigomycetes form zoospores that are posteriorly uniflagellate or polyflagellate. Members of the glomeromycota form arbuscular mycorrhiza, a form of mutualist symbiosis wherein fungal hyphae invade plant root cells, and both species benefit from the resulting increased supply of nutrients. All known glomeromycota species reproduce asexually. The symbiotic association between the glomeromycota and plants is ancient, with evidence dating to 400 million years ago. Formerly part of the zygomycota, the glomeromycota were elevated to phylum status in 2001 and now replace the older phylum Zygomycota. Fungi that were placed in the Zygomycota are now being reassigned to the glomeromycota, or the subphyla Incerticedes Mucoromycotina, Kixolomycotina, Bazupagomycotina, and the Entomopteromycotina. Some well-known examples of fungi formerly in the Zygomycota include black bread mold and Pylobolus species, capable of ejecting spores several meters through the air. Medically relevant genera include Mucor, Rhizomucor, and Rhizophus. The Ascomycota, commonly known as sac fungi or ascomycetes, constitute the largest taxonomic group within the Eumycota. These fungi form meiotic spores called ascospores, which are enclosed in a special sac-like structure called an ascus. This phylum includes morels, a few mushrooms and truffles, unicellular yeasts, and many filamentous fungi living as saprotrophs, parasites, and mutualistic symbionts. Prominent and important genera of filamentous ascomycetes include Aspergillus, Penicillium, Fusarium, and Claviceps. Many ascomycete species have only been observed undergoing asexual reproduction, but analysis of molecular data has often been able to identify their closest teleomorphs in the Ascomycota. Because the products of meiosis are retained within the sac-like ascus, ascomycetes have been used for elucidating principles of genetics and heredity. Members of the Basidiomycota, commonly known as the club fungi or basidiomycetes, produce myospores called basidiospores on club-like stalks called basida. Most common mushrooms belong to this group, as well as rust and smut fungi, which are major pathogens of grains. Other important basidiomycetes include the maize pathogen Ustilagomades, human commensal species of the genus Malassezia, and the opportunistic human pathogen Cryptococcus neoformans. Thank you for watching. Brought to you by WikiVD Documentaries. Please like and subscribe below. Please like and subscribe below.